Hello everyone, good morning. I'm Paulo Silva, a bioinformatician working at CGPP IBMs, and I'm going to tell you our adventure in over 6,000 exomes sequenced in our genetics laboratory and some bioinformatics pitfalls, challenges, and opportunities that we learned along the way. First, a brief introduction about us. As I mentioned, the Center for Predictive and Preventive Genetics provides clinical and laboratory genetics services to the community, as well as education and training. We are a part of IBMC, the Institute for Molecular and Cell Biology, and belong to the I3S consortium, which also includes INIB and Patimub at University of Perth. In order to provide clinical genetics testing services, we need to maintain a state-of-the-art laboratory practices as well as follow the standards recommended for genetics labs. We are a fully licensed medical genetics laboratory at the RERS, and we are accredited by the Portuguese Institute for Accreditation since 2014. We also participate in inter-laboratory external molecular quality assessments. Next-generation sequencing has revolutionized clinical genetics testing, since a single reaction has the ability to sequence millions of different fragments in a single run. This can range from amplifying a single exome or gene to sequencing the whole human genome. We perform whole exome sequencing, which is the capture and sequencing of the protein coding portions of the human genome, which harbors most of the known disease-causing variations or mutations. Briefly, NGS analysis and clinical reporting is divided into three major steps. The primary analysis, which consists of the actual sequencing itself on the equipment, followed by the secondary analysis, a bioinformatics step, which aligns the sequenced output or reads from the primary analysis with the human reference genome and discovers the differences or variants between that reference genome and the proband under analysis. This generates SPAM files containing those reads and the VCF file containing variants. Those variants are then annotated using expert-curated databases, which are then interpreted by clinical molecular diagnostics technicians and reported, called the tertiary analysis. Since we started this adventure in 2016, we have seen a major uptake of these exome-based genetics tests by physicians, shown here in this chart. And so far, we have sequenced and analyzed over 6,700 different patients. Now, I'm going to go over a few of the challenges and pitfalls that we have encountered during our secondary and tertiary analysis. One major issue of secondary analysis is the detection of false positive variants, as well as missing the detection of true positive variants, giving us a false negative result, which can have clinical implications. One example of a false positive is in the gene CYP21A2, which is associated with congenital adrenal hyperplasia and hyperandrogenism. In this example shown, pathogenic variant detected by whole exome sequencing was absent when confirmed by Sanger sequencing. Reads containing the false variant come from a known pseudogene CYP21A1P. Therefore, we perform Sanger sequencing of variants with known pseudogenes in order to eliminate variants coming from those highly similar reads. Another possible problem in variant detection is the presence of regions in some genes which have low coverage. For instance, in the gene ABCD1, which is located on the X chromosome, the last exons are not sufficiently covered, and in a patient with a negative test result and a very strong clinical suspicion of adrenal leukodystrophy, in the particular example of this gene, Sanger sequencing fill-in is performed to cover those regions. There are even regions with no coverage, such as in the case shown here of the SLC37A4 gene, which is associated with an autosomal recessive glycogen storage disease. However, a more recent capture kit that we use fully captures the exonic regions of this gene, highlighting that not all exons are born the same, and that improvements in capture kit technologies can improve diagnostic yield without the need to jump to whole genome sequencing. Segmental duplications are stretches of DNA up to 400 kilobase pairs that share a high sequence homology, up to 100% in some cases. One such example are the SMN1 and SMN2 genes mutations in which causes spinal muscular atrophy. Another example are mutations in PKD1, which account for around 80% of autosomal dominant polycystic kidney disease, and whose detection is complicated by the presence of 6 pseudogenes duplicating the first 32 exons of this gene. A peculiar example of a pseudogene is the protein kinase PRKRA, associated with dystonia. In one patient with early onset Parkinsonism and dystonia, we discovered that an heterozygous variant by whole exome sequencing reported in the literature to be disease-causing. However, to our surprise, Sanger sequencing confirmed an homozygous variant instead. Further manual expansion of the BAM file revealed a processed pseudogene whose reads were aligning with the functional gene's locus and making it seem as if it was an heterozygous variant. Searching the literature, this processed pseudogene is only present in some HLA haplotypes. Some types of variants are refractory to detection, namely insertion of mobile elements such as ALUs, deep intronic variants not covered by the capture kits, and repeat expansions. One such example is the trinucleotide CAG repeats expansion in exon 1 of HTT, which causes Huntington disease, and not autosomal dominant disease devastating to affected candidates. These types of expansions are not routinely detected using short reads and are ideally performed using targeted fragment analysis. Recent advances in bioinformatics software and algorithms can make it feasible to screen for them in whole genome sequencing data and even in some cases, whole exome sequencing data. For a long time, copy number variants were also a type of variation not detectable using whole exome sequencing. However, recent advances in algorithms and software now allow CMVs to be able to be included in standard exome-based genetic tests. Very briefly, the read depth of each covered region is calculated from the aligned reads in the BAM file and then statistically compared with other patient samples in order to discover if there is an increase or decrease in coverage, corresponding to a duplication or a deletion, respectively. You can see more information about CMV detection in our lab in a poster by Fatima Lopes, presented in this symposium. Using read depth CNV calling, we can detect large variations such as this one involving the short arm of chromosome 9, a trisomy involving 203 known genes in a patient with intellectual disability and epilepsy, among other features. We can also detect short CNVs spanning only one exon, but this case in particular was quite interesting as a partial deletion of exon 7 in the titin gene was not detected by the software we routinely use. However, manual inspection of the BAM file allowed us to discover the precise breakpoints of this short CMV, which is likely responsible for the phenotype of this patient with a complex polymorphomotive syndrome. Sometimes, the molecular diagnostics of some patients is behind two challenging variants, such as this case of a patient with multiple craniofacial anomalies that carried a heterozygous deletion in the TXNL4A gene. This gene is responsible for an autosomal recessive craniofacial disorder, but only one variant was present. It had no pair. Once again, manual inspection of the BAM file was key in discovering a second deletion in the promoter region of the gene, which was described in the literature and confirmed by Sanger sequencing. For more information on this particular case, please check the poster in this symposium by our physician, João Freixo. So far, we saw pitfalls and challenges in the variant detection step of secondary analysis. But many also exist in tertiary analysis, the annotation in the interpretation step of the NGS data analysis. One is the representation of transcripts between different databases, such as illustrated here for the gene PALM, where we can see that NCBI's RefSec and EmboDBI's ensemble and gene code transcripts have considerable differences between them. Care must be taken that the clinically relevant transcripts are correctly used, and projects such as locus reference genomics, or LRG, and matched annotation from NCBI and MBODI main are attempting to resolve these inconsistencies. Differing transcripts of the same gene or isoforms can be expressed in different tissues or in different phases of development. Here, a variant in the CDKL5 gene, which is known to cause an epileptic encephalopathy, was found in a newborn with focal epilepsy. In the canonical transcript, this variant was infound. However, in an alternative transcript, which is expressed in the brain, this variant causes a frame shift. A nearby similar frame shift variant is described in the literature as causing early onset seizure, which is compatible with the clinical presentation of this program. Another well-known example is the gene encoding titin, which expresses different isoforms in cardiac and skeletal muscles. Allele frequencies from large population databases such as the 1000 Genome Project, EXAC, and NOMAD have helped tremendously in filtering common polymorphisms and allow the reduction of the number of variants that tertiary data analysts and molecular geneticists need to interpret. However, Portuguese population-specific variants are absent in these major databases that I mentioned, and building such a database will further reduce the number of variants that need evaluation and improve the speed of interpretation. Modes of inheritance in genetic disorders can also change as more families are diagnosed and additional studies performed. An example is spastic paraplegia 7, which for a long time was considered autosomal recessive, requiring biallelic pathogenic variants for the disease to manifest. However, there is no evidence that autosomal dominant transmission is present in some families. Finally, there can be some zygosity issues in the case of overlapping variants. In this example, an homozygous single nucleotide variation in the gene MFS-D8 overlaps a duplication of the whole gene. Care must be taken in interpretation and reporting of this variant to account for the presence of 3 alleles instead of the expected 2. The many pitfalls that I mentioned during this presentation can be mitigated by implementing improvements in bioinformatics pipelines used for detection and annotation of clinically relevant variants. The challenging aspects also serve for training our data analysts and molecular diagnostics technicians, as well as other trainees, and help build NGS literacy. With such a large cohort of over 6,000 patients sequenced, a Portuguese variant database will help accelerate and improve diagnostics and reduce turnaround time. And this dataset can also serve as a tool to group together undiagnosed patients and find patterns and signatures that can allow the discovery of new gene-disease associations. In conclusion, NGS, and in particular exome sequencing, was quickly adopted by physicians for diagnosing rare genetic disorders. Their clinical utility is currently unquestioned, and are now being considered and recommended as a first-tier diagnostic tools for pediatric patients with congenital anomalies or intellectual disabilities. The limitations described here should be clearly presented to clinicians, and good bioinformatics practices implemented to mitigate false negative results and therefore improve diagnostic yield. Our large dataset of over 6,000 exomes can be leveraged to improve the precision of genetic studies and is an important asset for applied research of patients with undiagnosed rare disorders. Our tale of exome-based genetic tests continues just as new technologies enabling more comprehensive and demanding tests are coming to the clinic in the very near future. Finally, I would like to acknowledge all of CGPP team members, past and present, and in particular wet lab and dry lab teams, as well as our directors. Thank you.